Hey guys, I thought we'd pick up with reading guide number 3, which is all about the Calvin cycle. And so let's start with the first question that asked, what original reactant of photosynthesis is used in the Calvin cycle? What's the actual product of the Calvin cycle, and what from the light reactions used in the Calvin cycle? And guys, this really goes back to this overall kind of description of the two parts of photosynthesis. We got the light reaction and we've got the Calvin cycle. We know that CO2 is used in the Calvin cycle because we're making the glucose. And so at this point, we've got to have that carbon coming in. ATP and NADPH are coming over from the light reaction. It's where we're going to get our hydrogens to add to our CO2 to get our glucose. And so just kind of remember that overall. And really, that explains that first question. The actual product is glucose, technically G3P, which we're going to get into that a little bit more here in a second. But ultimately glucose through the combination of this molecule called G3P. So let's go ahead and move on to question 2. And so to do this, you guys have a model of the Calvin cycle, and I'll show it to you completed here in a second. But let's just go ahead and draw it with me, okay? Go ahead and humor me here. So we got the big Calvin cycle, and we can go ahead and just label it Calvin cycle here. Okay. And we're going to start with this molecule abbreviated RUBP. Okay. It has a long name that you've probably seen in your reading, ribulase biphosphate, but we're going to call it RUBP this whole time. And RUBP is a 5-carbon molecule. Okay. And there are actually 3 of these that are going to be used in the Calvin cycle. And so we kind of start with this RUBP. Now we know that CO2 is what's coming into the Calvin cycle. Okay. And for each circle of this, we're going to need 3 of those. And this is, going to make kind of 3 turns to make our G3P here in a second. And so you might even want to put a 3 out here. And it's going to take 2 of these to ultimately to make our glucose. And so what happens is the CO2 comes in and the carbon bonds with the RUBP. Okay, so kind of right off the bat, a 6-carbon molecule is produced. And that 6-carbon molecule is very unstable. Okay, and this is technically the carbon fixation part of the Calvin cycle. Okay, this would be phase 1. And this molecule right here is very short-lived. It just won't stay. Okay. The enzyme that makes this combination is called Rubisco. And that's the enzyme that combines the carbon with the RUBP. Rubisco, we know, is the most numerous enzyme in chloroplast. And actually, they believe this is the most numerous protein in the world because it's used for this process so very much. And so to our knowledge, Rubisco is the most numerous protein, period, in the world, which is really saying something. And so what happens right off the bat, and we've seen this before, is that we now have 3 molecules of this 6-carbon compound, and it breaks apart into 6 3-carbon molecules. Okay. And that's that phosphoglycerate. And that phosphoglycerate is almost immediately phosphorylized. And so we have 6 molecules of ATP that become 6 molecules of ADP. And so, like we've seen before, we have a phosphorylated molecule here. And then it's going to process some more and actually get some energy from NADPH which is going to become NADP. And there's going to be 6 of those as well. And we're going to have an even farther processed carbon molecule. And we still have 6 of those. Okay. And now we've really entered into phase 2. So carbon fixation was phase 1. We've entered into phase 2, which is reduction, because we're adding some electrons now. Okay, so reduction as in redox reaction. And ultimately, through this, we're going to make the plant is going to make a 3-carbon molecule, 6 of these, called PGAL. Okay, going to be abbreviated PGAL. And for most of these, the abbreviation is really what I'm worried about you knowing, okay? And what happens is that one of those PGALs is going to come out of the cycle. And at this point, it's going to be called G3P. And this is really the output of, let me move that up a little bit. That's the output of the Calvin cycle, okay? And G3P is ultimately going to combine with another G3P to make glucose. So that is where that glucose is going to come from. So one of these was able to break free, but that leaves us with 5, right? PGALs. So we have 5 PGALs now, and they are going to get a little shot of energy in the form of 3 ATP that become 3 ATP to get us back to ribulose biphosphate, which was what we needed to kind of get this cycle started in the first place. Okay. And so we see kind of these 15 carbon molecules use this input of energy from 3 ATP to rearrange themselves into 3,5 carbon molecules again, which can combine with CO2 through the help of Rubisco to come back down and release this G3P, where we can have a molecule of glucose ultimately. And so that, guys, is the Calvin cycle. Let me go through it one more time. So we start. We've got to have RUBP in place. It's a 5-carbon molecule. There'll be 3 of them for a total of 15 carbons. The enzyme Rubisco, most numerous protein in the world, helps CO2 bind to that RUBP, making a 6-carbon molecule, very short-lived, 3 of them ultimately. It's going to break down into 3 carbon molecules. We've seen this happen before. It's the carbon fixation part. We're going to have an input of 6 ATP to phosphorylize it. We're going to have an input of 6 NADPH, help rearrange it. This is the reduction part where it takes on these electrons. So ultimately we have these PGAL. One of those is going to exit as, G3P, which can combine later with another G3P to make glucose. That PGAL comes back up here, and this is phase 3. We need to add to your notes. This is phase 3, which is the regeneration of that CO2 acceptor. And that CO2 acceptor is RUBP. And we get back up here, takes 3 ATP, we get RUBP back, and we can start all over. Now remember, guys, when we were talking about the cyclic versus the non-cyclic, one of the things that comes into play here is that we've used 9 ATP, and we've only used 6 NADPH. So, if this continues, we're going to run out of ATP, right? And so it's going to go back to the light reaction and need to have some cyclic electron flow to regenerate any missing ATP. And this is why there's that variance between cyclic and non-cyclic and how much of each of these molecules we use. Here's the picture that goes with your paper. I would like a little more detail, more like what I drew out. Okay, but if you want to kind of pause the video and make sure you've got everything filled in, or kind of, I guess, make sure, check yourself. Okay, those are the basics of the Calvin cycle. So if we go through these questions, let's kind of take a look at these. In carbon fixation, what atom is carbon added to? Okay, well, it's added to RUBP, right? In carbon fixation, carbon would be added to RUBP. What enzyme is responsible for that? Rubisco is responsible, right? The most numerous enzymes responsible. What are the products at the end of carbon fixation? At the end of carbon fixation, just this part of it, you've got 2 3-carbon molecules. You got 6 total because we're talking about 3 turns, right? But you've got these 3 carbon molecules. In reduction, which is this part, what is added to the 3 carbon at the beginning of reduction? Well, phosphate from ATP, right? It gets phosphorylated. What's the product of this phase? 6 molecules of PGAL or G3P. And one of those is going to leave the cycle. The other 5 are going to keep going. Okay. What's the ultimate product of phase 3 of the Calvin cycle? This is phase 3. The ultimate product here is RUBP. We're getting back to that 5-carbon molecule that's going to be the CO2 acceptor in this process. And how does the light reaction sustain the Calvin cycle? Well, we've got to have this ATP and we've got to have this NADPH, right? And so when we go back up to this kind of overall picture, the light reaction's constantly sending ATP and NADPH to the Calvin cycle to keep it going, because the Calvin cycle is really the payout of this glucose. And guys, I think that kind of gets us through this reading guide and the Calvin cycle. If you have any questions for me, please reach out. Hopefully this is making sense to you at this point. One thing I want you to do today is on page 202, which there are extra books in the room, so you'll probably have time, but you could do it at home too. At the bottom of page 202, I want us to do this concept 10.4. Okay, concept check. There's 5 questions. I want you to do all 5. You can write it on a piece of paper or type it out and turn it in. I'll put a place on Classroom to finish it. This is just a good kind of stop point to check and say, okay, are we getting this? Are we getting the Calvin cycle? So I want you to do concept 10.4 check today in class and turn it in to me, okay? And we will move forward with the next reading guide tonight. This is going to be reading guide 4. I'm going to post it as well. There's 7 questions. Or actually, I think there's 8 questions. Let's work through the first 3, okay? And then we'll kind of go from there tomorrow. So, I know you guys have the ACT, the juniors have the ACT tomorrow, but I will be making a video for you, so I still will need you to get on and do some work for AP Bio in the afternoon, okay? So, hope you're having a good day. And I hope to see you all soon.